So just to refresh your memory on what we're talking about is the goal is to figure out if the inheritance pattern of one gene affects the inheritance pattern of another. So the idea is because round and yellow came from the same parent and wrinkled and green came from the same parent, when you cross them together and make a heterozygote and then cross those heterozygotes together, does the fact that round and yellow came together and wrinkled and green came together influence the likelihood of them staying together in that last generation? So are you more likely to keep big R's and big Y's together and little r's and little y's together? And that's the goal of this cross. So I know that this is a terrible drawing, and I take at least partial responsibility for it. But can you guys at all see the letters on this? Like, can you see that that's big R's and big Y's and little r's and little y's? Can you tell that that's a wrinkled P? Can you tell if that's a round and yellow P? Okay. So what we just talked about was taking homozygous round yellow and crossing them to homozygous wrinkled and green. And we said you get the heterozygous type. They're big R, big Y, little y. Okay? So now what we want to ask is when you cross together two heterozygotes, what do you get as offspring? And so Mendel did this cross. And when he did this cross, what he got, and remember, he doesn't know about meiosis, but what he got was 9 parts round and yellow offspring, 3 parts round and green, 3 parts wrinkled and yellow, and 1 part wrinkled and green. So 9 parts round and yellow, the dominant for both types, 3 parts round and green, dominant for one, recessive for the other, 3 parts wrinkled, yellow, recessive for one, dominant for the other, and 1 part wrinkled and green, recessive for both. And based on that, those numbers, 9 to 3 to 3 to 1, he said that the traits do not influence each other's inheritance. And we're going to walk, to me, just looking at the numbers, I would not be able to figure that out myself. But when he did actually figure it out just from those numbers. But what we're going to do is we're going to walk through meiosis and show you why that makes sense. All right? And you're going to recover, hopefully, at the end, a 9 to 3 to 3 to 1 ratio after you make a mega Punnet square to cross together these two heterozygotes. So the first thing that has to happen whenever the meiosis is going to occur is we have to make our sister chromatids. So we take our big R's and our little r's and we double them. We take our big Y's and little y's and we double them. We make our tetrads. Everybody see that? And then the tetrads, and you've drawn this so many times that I hope that this is pretty straightforward. This pair of homologous chromosomes of the red and blue type are going to move over, the 2 sister chromatids of each, into one cell. These 2 will move over and be joined together in another cell, and then they'll line up and split. That's just regular meiosis. But when they line up, the 2 tetrads are totally independent from each other. They don't associate with each other. The red and the blue, different chromosomes altogether. So when they line up in meiosis, it's a 50-50 shot that they line up with the big R's on the same side as big Y's and little r's on the same side as little y's, or big R's with little y's and little r's with big y's. It's like, I don't know if this will help, but if you imagine like the 2 tetrads are my fingers here, they either line up in the middle of the cell where the pairs of, they either line up in the middle of the cell where the chromosomes that contain the dominant types are on the same side for both genes, or they're just flipped. So the dominant type of one now is lined up on the same side of the cell as the recessive type of the other. And it's just a random thing which way they happen to orient. And if you incorporate homologous recombination into this 50-50 shot, you end up getting the same result as we're talking about here. So we're not going to really bother sketching recombination. But if you were to incorporate it, you end up with the same outcome. Okay. So if big R's line up with little y's, suddenly we have a cell with big R's and little y's together, and a cell with little r's and big y's. Those then split, giving you gametes that are little r big Y and big R little y. So every time a cell, have you ever seen those games, you know, it's like a marble and it drops down and then it keeps changing direction as it goes down hitting pegs? Well, it's kind of like that. When the cell drops down in meiosis, it either drops this way and ends up making big R's with little y's and little r's with big y's, or it drops and flows this way and gives you big R's with big y's and little r's with little y's. So each time a cell does meiosis, it kind of goes one way or the other. So if you take an organism and let it do a bunch of meiosis 1,000 times, what you end up with is an equal ratio of these 4 different types of gametes. So look at the genotypes of those gametes. Big R's with big Y's, 25% of the time that's what you get. Little r's with little y's, 25% of the time that's what you get. Little r's with big Y's, 25%. Big R's with little y's, 25%. So when you set up a Punnet square, what you're going to do in this case is a little different than how you set a Punnet square up previously. So if this is my Punnet square, who is parent 1? What's their genotype? Big R and big Y, yes, but what else? Little r and little y. So by convention, we group the 2 alleles for the same gene together. So we would write the genotype of the first parent, big R, little r, big Y, little y. And who are we crossing it to? Itself or the same genotype. So parent 2 is also big R, big Y, little y. Now I just told you in meiosis, when a heterozygous individual for 2 different genes that are on different chromosomes goes through meiosis, they make 4 genotypes of gametes all at equal frequencies. So on your sheet, what I imagine happens next is in these 4 lines here, you write those 4 possible gametes that can be produced. You guys see where I say, next make these plants go through meiosis. What kind of gametes do they make? And this is the meiosis-based explanation for the type and frequency of gametes they produce. Does it make sense? Like, one thing that a lot of people get confused by is they think that one cell does both of these. And that's not the case at all. It's one cell either lines up with big R's and big Y's together, and little r's and little y's together, or it lines up in the opposite orientation. So what are those 4 gamete types? Take it away.
Big R, little y.
Big R, little y.
Little R, big Y.
Little r, big Y.
Big R, big Y.
Big R, big Y.
And little R, little Y.
And little r, little y.